And finally, for the endocannabinoid section, I'd like to introduce Dr. Danielle Piamelli, who's going to discuss the therapeutic potential of the endocannabinoid system. Danielle's coming from the University of California, Irvine, where he's the professor of anatomy, pharmacology, and biochemistry, and he is the Louise Turner Arnold Chair at that department. Dr. Piamelli has been interested in arachidonic acid for a lot longer than cannabinoids were around, endocannabinoids were around. He was working on icosanoids before anandamide was discovered, but jumped in completely into the FAAH when FAAH was discovered, and has done a great deal of work on FAAH, and has also done a lot of work commercializing FAAH for therapeutic potential. He has 25 patents and 3 startup companies. So I'd like to introduce Dr. Piamelli.
Good morning. I'm honored to be here. As our thoughts go to the victims of the terrorist attack, I'd like to remind ourselves that we can do good things, and evidence-based medicine is one such thing. I've been charged with the task of going over the therapeutic potential of the endocannabinoid and the cannabinoid system at large. You have heard from the previous speakers that the endocannabinoids can regulate a variety of fundamental physiological functions. The speakers that preceded me have focused their attention on the brain, but really the entire body is a target for the endocannabinoids. I have divided my short presentation into 2 blocks. The first one will cover the cannabinoid receptors. You have heard a lot already about from Dr. Mackey. And the second one will cover the 2 main endocannabinoids that were introduced to you by Dr. Cravat, anandamide and 2-AG. So let's start off with the cannabinoid receptors. These, as we have seen, are G-protein coupled receptors, and they're present in 2 established subtypes, CB1 and CB2. They activate a rich repertoire of intracellular transduction mechanisms Dr. Mackey alluded to. The CB1 receptor is certainly one that we are most interested in. Dr. Mackey showed us some very beautiful pictures of its localization in the brain. I would like to stress, however, that this receptor is truly ubiquitous in the sense that it is expressed by many different cell types in the body, not just by neurons, not just by astrocytes or microglia, but also by hepatocytes, by adipocytes, and a variety of other cell lineages. The CB2 receptor, on the other hand, is a little bit different because it is mainly expressed by cells of the immune lineage, either innate or adaptive immunity, including macrophages, B lymphocytes. So in a sense, CB2 receptors also are ubiquitous insofar as macrophages are ubiquitous. But they are ubiquitous in a different way as the CB1 receptor is. Let's start with the CB1 receptor, the one we actually know the most about. And what can we do with this receptor? Well, we can do a number of different things, but the first one is clearly to try and activate it and see what therapeutic benefit we could achieve that way. And this is done with what in pharmacological lingo is called a receptor agonist. Now, receptor agonist is a broad term that encompasses different animals, really, quite different animals. There are such things as partial agonists, and Dr. Mackey gave us a wonderful explanation about how a partial agonist work. But let me just go over this very quickly again. It is an important concept. So if one looks at the effect size of an agonist, so one sets that a receptor can produce a response that is 100%, then a partial agonist would produce a 60% or a 70% or a 20% of their response. And this differentiates, therefore, a partial agonist from a full agonist. The full agonist will produce that maximal effect I mentioned before. So it is interesting that the two endogenous cannabinoids, anandamide and 2-AG, are different in that anandamide is a partial agonist, whereas 2-AG is a full agonist. It is also, I think, interesting that THC, the main psychoactive component of cannabis, is also a partial agonist. Now, I like to introduce you a new term, which is not really new, but I didn't make this up, but it's a term that I think we should keep in mind, a superagonist. Compounds such as CP55940 in given situations can actually cause a greater than maximal effect. So it becomes kind of a relative, saying what is full and what is super becomes almost like a relative term. But to give you a sense again that if we create probes, we can really change the response in ways that nature did not anticipate. And this is what we have done, I think, with a lot of these cannabinoid agonists, activating CB1 receptors a lot more than anandamide or 2-AG were meant to do by evolution. Finally, but not unimportantly, we can modulate the receptors through allosteric means. And again, Ken has given you a very nice introduction to this. An allosteric modulator really has little or no effect by itself, as you can see here. But if now a ligand, for example, an endogenous ligand comes into action, then the actions of this ligand will be enhanced by the allosteric modulator. So all these things are not theory. All these things have been done. So what have we learned by using in the lab, in animal models, in vitro models? What have we learned from using these different type of receptor agonists? Well, of course, we have learned a lot about THC. There are decades of work on THC, which is a partial agonist. We know that THC is already a therapeutic agent. It is sold as marinol, but it's also present in cannabis, and it's probably the most important cannabinoid present in cannabis. The commercial cannabis that is found in the streets or in many stores dispensaries, is mostly composed of THC at pretty high concentrations. So it is a therapeutic agent already. It does produce CNS-based side effects, and this is due to the fact that the cannabinoid receptors, or CB1 receptors, are really widespread throughout the brain. So some of the things it does, for example, we have heard the analgesic effect, we have heard about the appetite-stimulating effect, the anti-spasticity effects. Those are useful effects, but they are inevitably accompanied by psychoactivity, by loss of memory, by loss of motor coordination. It is the nature of this drug to produce those effects. The two things cannot be separated using this type of drugs. If we look at full agonist or superagonist, this problem is magnified. These compounds are really hard to work with. They do have very interesting pharmacological actions, but they also inevitably activate all cannabinoid receptors and cause CNS-based side effects. Where we have more leeway is in the area of allosteric modulation, and this is an area that requires more work, but it's a promising area because, again, when we modulate allosterically a receptor, we are only making this receptor more sensitive to the endogenous ligand. So you have to picture this. You're not activating receptors that are not normally engaged. You're only activating, if you wish, enhancing the activation of those receptors that normally see their ligand. So you're essentially magnifying an endogenous action. That is why I believe this area is very promising and needs to be explored. Now, because it is so hard to work with direct-acting cannabinoid receptor agonists, CB1 receptor agonists, people have started to play around with an alternative possibility to make drugs that do not enter the CNS. Since the majority of the side effects of CB1 activation are in the brain, a logical way of preventing that is just making compounds that don't pass the blood-brain barrier, that act exclusively in the periphery. And there are companies and scientific and research labs that have worked on this. The results have been, however, disappointing because at least with the few drugs that have been explored, these drugs have shown peripheral side effects mediated by the cannabinoid receptor itself. As I said before, CB1 receptors are ubiquitous, and they're involved in the regulation of metabolism, particularly lipid metabolism, as well as feeding. So one common side effect of peripheral agonists has been the stimulation of feeding, the increase in body weight, as well as these metabolisms that create problems over a long period of time. However, I would like to say that this is the jury is still out there when it comes to the side effects produced by these compounds, because we don't have enough of a body of knowledge yet. So that's an area that I think still deserves consideration. When we look at CB1 receptors, and agonists at CB1 receptors, we must consider the dark side of CB1 pharmacology, which is particularly represented by the superagonists that compose such abused substances as spice, K2, etc. These are compounds that typically overly activate CB1 receptors, and of course they are being now abused by a substantial number of people, particularly our youth. About 6% of 12th graders in 2013 used spice or spice-like compounds, and their impact cannot be underestimated. So what about blocking the cannabinoid receptor? Clearly we can do this, and we do this with what pharmacologists call antagonists. But again, just like agonists, they come in multiple flavors. There are neutral antagonists, there are inverse agonists, and they are allosteric or biased modulators. So what are these things? Well, neutral antagonists just block the receptor, easy. Inverse agonists seem to do something different. As Dr. Mackey explained, they actually force the receptor into a conformation that is negative, so the receptor becomes actively inactive. The allosteric or biased modulators are very interesting compounds, and again, Dr. Mackey explained them. Just let me give you again the gist of what they do. If, say, CB1 receptor is coupled to two signaling pathways, 1 and 2, okay? There are more than that, but let's say it's only 2. Well, what a biased modulator would do, typically, is just silence out one of those pathways. And if we are lucky and that particular pathway is the bad pathway and the other one is the good pathway, then we got potentially a drug. So what have we learned by using these different compounds? Well, we have learned a lot, but I think there is still a lot to be learned. We have used inverse agonists in the clinic. They've been extensively studied in animal models and then in people, particularly for metabolic diseases and obesity at the center of those diseases. We know that these compounds are effective in obesity. We know that they decrease body weight, they improve the overall metabolic panel, and they do so in a quite effective way. However, we also found that they exert CNS side effects. These CNS side effects are linked to the important role that the endocannabinoids play in regulating anxiety, in regulating mood and affect. Particularly anandamide has been involved in this, in such role, and blockade of anandamide is responsible, I believe, for most of the CNS side effects of these compounds. What about neutral antagonists? Neutral antagonists are, of course, more moderate in their action because they do not force the receptor into an inactive state. They kind of block the receptor prevent the receptor from being activated. And there are studies ongoing on neutral antagonists, but we still have preciously few such compounds, and more work is needed there. What about allosteric and biased modulators? Again, here we have very promising data, and Dr. Mackey alluded to a compound, pregnenolone, which has been now optimized for the treatment of cannabis addiction. A few important thoughts about what we have learned from CB1 antagonists in metabolic syndrome. I would like to point out that therapeutic effects in human metabolic syndromes were predicted by animal studies, and the side effects also were predicted. And this is important because it gives us confidence of the predictive value of our models, so that as we test new drugs, we should believe in what we see and not just be acritically skeptical. One such drug which I think deserves attention is peripheral CB1 antagonists, which have been shown to be highly effective in animal models of metabolic disease and deserve further study in humans as well. The CB2 receptor, with which I move now, is, to paraphrase Oscar Wilde, a sphinx wrapped in a mystery. We have seen that they are highly expressed in immune cells. Their function, however, are unknown. Activation of this receptor is analgesic, anti-inflammatory, and tissue protective, but the mechanism of action is unclear. But most importantly, we do not have good probes to study this receptor, and therefore more work is needed before we can definitely describe their actions. So in the next couple of minutes, I'd like to go over anandamide and 2-AG. To start off, I'd like to give you a sense of what personally I think these compounds do based on an assessment of the literature over the last several years. 2-AG, we have heard, is the workhorse. Dr. Cravat used a very nice metaphor. Indeed it is. Another way of looking at it is that it's a point-to-point messenger. It goes from one side of the synapse to the other retrogradely. Instead of going from pre to post, it goes from post to pre and exerts many functions in the CNS as well as the periphery. Anandamide, instead, is more of a modulatory transmitter. It acts like what people like to call a volume transmitter, and has responses, an array of responses that are linked to the control of stress in the central nervous system and pain in the periphery. So how can we use these signals for therapy? Well, let me remind you, first of all, how these compounds are naturally produced. They're produced from lipid precursor in cell membranes. Dr. Cravat showed some structures. I decided that I wouldn't show you any structures, but the idea is the same. We got the lipid precursor converted into biologically active endocannabinoids, and then metabolites. Metabolites that can be active or inactive, but be what it may, if we block the biological transformation of these endocannabinoids, then we can enhance the system's intrinsic regulatory functions, possibly with greater selectivity than just activating cannabinoid receptors indiscriminately. This can be done by using inhibitors of 2-AG degradation, which target three main enzymes. They're called mannose-glycerol lipase, Dr. Cravat described it, ABHD6 and COX-2. And these compounds have been shown in animal models so far to do a number of interesting things, which are listed here. But these compounds also have non-endocannabinoid functions, which include cancer suppression, reduced neuroinflammation, etc. Relatively mild side effects. The inhibitor of anandamide degradation prevalently target the enzyme FAAH, fatty acid hamatohydrolase, and are effective in a number of animal models, but especially in human cannabis abuse, has been shown recently, and they have a very mild side effect profile in animals and humans. I would like to point out that we know a lot about the safety of these compounds. 7 FAAH inhibitors have been tested, 18 phase 1, phase 2 studies have been conducted, over 1,000 subjects have been seen, no serious side effects have been noted so far. There has been recently a very tragic event in France about a compound called BA102474. And what do we know about this compound that can explain its unusual toxicity? I don't have the time to really go over this carefully. I'd like just to point out that it's a weak FAAH inhibitor. It's got an unusual chemical structure. It's a poorly characterized target selectivity, very limited animal pharmacology, animal toxicology showing lethal effects at high doses, and an unfavorable human pharmacology and human pharmacokinetics, with toxicity occurring in humans at doses that are consistent with enough target effect. So what we conclude about these terrible events, that we must shed light on many aspects of the actions of this compound, but the toxicity was most likely due to enough target effect that it was not mediated by FAAH, and that much greater caution should have been exerted before and during the clinical testing for this compound. And I'd like to remind all of us that guilt by association is not sound legal doctrine. Therefore, we should not stop research on FAAH inhibitors just because one compound turned out to be a terrible molecule. So looking forward, what direction should we go to? Certainly, I believe allosteric positive CB1 modulators offer a promise. Neutral CB1 antagonists also offer promise. Likewise, peripherally restricted CB1 antagonists, particularly in obesity and metabolic syndrome, offer promise. And both global and peripheral FAAH inhibitors offer promise. Global inhibitors for anxiety, post-traumatic stress disorder, autism spectrum disorder, and a peripheral FAAH inhibitor for nociceptive pain, for reasons that I didn't have the time to go through today with you, but there are animal data supporting it. Also, MGL inhibitors, or MAGL, as other people like to call it, and ABHD6 inhibitors, which target 2-AG degradation, also have promise, particularly in epilepsy, pain, and cancer. Last but not least, I'd like to mention compounds that selectively inhibit the conversion of endocannabinoids to metabolites via the COX pathway. Again, I didn't have the time to go over them, but these are interesting molecules. And with this, I am done. The light is red, and I thank you for your attention.